Thanks for allowing Bluebird to present here on the story. We are a company that is very focused on making hope a reality for a number of patients with severe genetic diseases. I will be making forward-looking statements. As a company, we're really anchored culturally and practically in understanding the patient experience across all of the diseases that we so desperately seek to treat. And it's represented here by some of the images. We just last week had a couple of patients in from adrenoleukodystrophy and sickle cell community representing their plight. And it really is something that's foundational to the people we hire and to the products that we're ultimately trying to develop. Ethan is up there on the left side of the screen. And he's up there for a reason. He's an anchor for us. We got to know Ethan's family quite well. And he represents, unfortunately, somebody who didn't make it, who had adrenoleukodystrophy. And he's a constant reminder that we have to do better. The great news is that we have the tools, as being evidenced here today by all the great presentations. We have the tools and the technologies now to hopefully have no more Ethans out there, and that's ultimately certainly our goal. So we have two foundational platforms. One is based on the historic roots of the company in severe genetic diseases, and there we're targeting hematopoietic stem cells, ex vivo gene therapy, targeting those stem cells to gene modify the patient's own cells. We also have a platform in immunotherapy. Here our target cell is T cells, genetically modifying a patient's own T cells to affect disease in oncology and immunology. It's all, however, based on the last 5 years of investment, and significant investment at that, in the platform. And the platform really covers three different areas. One, lentiviral gene delivery, building a very potent, pure, reproducible, and scalable product. A manufacturing infrastructure that allows us not just to treat one patient, but many patients, and on a global basis, US and Europe currently, and plans to expand beyond that. And then we recently acquired a gene editing technology company that forms the foundation of our pipeline going forward. We all know this slide, which is that the field has been growing rapidly and exploding in the gene therapy world, whether it be AAV or lentiviral vector therapies, in the immuno-oncology world, whether it be CAR-T or TCRs or beyond that, and then certainly in the gene editing field. We at Bluebird have brought together technologies in each of those fields and built expertise and capabilities there, such that we think that we're building an integrated product platform that affords us great opportunity for a product engine going forward. That's represented in this slide, which really shows our pipeline overview. The first two products are our HSC platform products. We have two late-stage programs targeting childhood cerebral adrenoleukodystrophy and beta thalassemia major, which is the severe population of beta thalassemia. And then we have an earlier emerging program in clinical development for sickle cell disease. Beyond that is our immuno-oncology pipeline anchored in our BCMA program with Celgene. The first three programs are owned worldwide by Bluebird, and then the oncology program, we have some that are partnered and some that are owned 100%. We made great progress in 2015, as you can see here. Really, it was a year of generating some clarity around data, so early data across our beta thalassemia program, as well as our sickle cell program. But really, it was anchored in that data presented to regulatory authorities to get some clarity about how we can pursue registration with these programs. And you can see that we gained some clarity through conversation and dialogue with both EMA and US around the beta thalassemia program. I'll talk about that in a second. We've also shifted our immuno-oncology strategy where we have a much more independent approach here. We still are very excited about our relationship with Celgene, pursuing BCMA, but we also have a more independent set of programs behind that. And we started presenting data across all of our programs, built out the management team a bit further, and certainly helped fund the company with nearly a $500 million offering fairly recently. 2015-16, if 15 was a year of early data and regulatory clarity, 15-16, so the next 15 months are really about data and maturing data and larger sets of data. Presenting that data, as you can see, across multiple programs. For the first time in 2016, we will present on CCLD program, which is the phase 2/3 program. That'll happen in the first half of 2016, but also continuing to show data across our other programs as the data emerge and as the opportunities present themselves. We are making great progress on enrollment, but we'll continue to make great progress throughout 2016 in really finalizing our current trials and starting new ones, and then clearly advancing our pipeline with the gene editing technology and other technologies. So switching to beta thalassemia is the first program I want to touch on. We have two ongoing programs, clinical trials, one called the North Star trial, which is a global multi-center trial, phase 1/2. This trial, along with HGB-205, which is our French trial, which has both beta thalassemia major and sickle cell patients. It's upon this basis that we're seeking conditional approval in the EU, and I'll talk about that in just a second. But we expect to complete enrollment by the end of this year in each of those trials. Here are the data that we presented publicly that underlie that rationale for seeking EU conditional approval. What you see here, and we presented this in the past, on the left-hand side you see the patients. So those are individual patients across the two studies I just described, so two per study. And then as you progress from left to right, you're looking at the progression of transition of hemoglobin expression. On the left you see lots of blue. Why do you see that? Because that's the transfused blood that the patient was relying on to stay alive. And as you progress then out to 15 months for some, you see that blue disappear. They're coming off of transfusions, and what is showing up are the other globin chains. So you see red emerging. That is the globin production from the cells that were transduced and returned to the patient's body. These are their own cells, gene-modified, returned to the body, now reside in the bone marrow, that are now producing what is endogenous globin. They no longer need transfusions. You can see patient-to-patient variability. So you see from 3.8 at 6 months to 9.7 at 15 months. We should absolutely expect that. We expect that going forward. The great news here is that all 4 of these patients ended up being transfusion independent at the time points you see here on this chart. So that's great reason to believe, and that the impact on these patients' lives, we believe, is going to be tremendous. However, we've got to continue to deliver the data across all patient types, all genotypes, and with greater numbers. And that's what we intend to do through our regulatory strategy. So we believe, based on interactions with both EMA and FDA, that we have a path in the EU and the US, both accelerated paths. In the EU, it's through the Adaptive Pathways Program, where we have general agreement through discussions to pursue conditional approval based on the 2 studies and the data I just showed. Obviously, more data as those data emerge. That will be followed by full approval based on 2 other studies we plan to initiate here shortly, the HGB-207 and 208 trials. Those 2 trials, 207 and 208, will form the foundation for potential approval in the U.S. in an accelerated manner. So it's all these pieces are coming together and came together in 2015 in terms of providing us with some regulatory clarity going forward. Here are those 207 and 208 trials. So these are targeting beta thalassemia major, so the severe population of beta thalassemia major. They're global, single-arm, open-label trials. 15 patients in each, so a total of 30 patients. And the difference between the 2 trials is really the patient population. One is targeting adolescents and adults, and the other is targeting pediatrics. So that's an overview of the data, the regulatory path, and our plans going forward for beta thalassemia major. We also have a really exciting opportunity in sickle cell disease. And what you see here is the rationale for why we think the beta thalassemia data could be predictive of an outcome in sickle cell disease. I'm going to point 2 things out here. There's a lot on this slide, but I want to point 2 things out. One, our lentiglobin product is being used for both indications. And the reason for that is in the gene insertion, there was a single point mutation introduced, and that single point mutation confers anti-sickling properties. So there's a dual effect of lentiglobin and sickle cell. It's functioning hemoglobin, which they need, plus anti-sickling properties. This is the same mutation that's found in fetal hemoglobin, which is also known to have anti-sickling properties. And for our therapeutic, we've shown this in preclinical studies. The other reason to believe here, aside from the theoretical here, is that there is a condition that gives us a sense of what our target level should be for achieving therapeutic effects in sickle cell disease, and that's represented by hereditary persistence of fetal hemoglobin. It is what it sounds like. Sickle cell patients who have hereditary high levels of fetal hemoglobin, and what's been shown there is if they get approximately 30% of their globin production coming from fetal hemoglobin, they can take a full-blown sickle cell patient and make them phenotypic normal. So that's why, amongst other things on this slide, we believe 30% threshold, being above that, gives us a chance at having a dramatic effect in sickle cell patients. Obviously, the clinical data have to prove that, but that's the rationale. And what we're showing here are those 4 patients in beta thalassemia that you just saw, plus the sickle cell patient, which is the purple line on the very bottom, or the purple patient, subject 1204. And what's shown here is the percentage of globin production coming from the therapeutic gene that was inserted into the patient's cells. And what you see is every one of those patients at 6 months is above that 30% threshold. In fact, the range is 40 to 80%. So we believe, to be shown in clinical data, that this gives us a great opportunity to pursue sickle cell disease. And here is the first patient ever treated in sickle cell disease. And what you see is the same progression of transfused blood declining, red increasing, and you end up with about 50%, but 45% of combination of fetal and transduced globin that gets us, again, above that threshold. So we're very excited about our sickle cell program. This is a little bit more data about that patient. He was transfusion dependent, had a hospitalization history, but no hospitalization since infusion with our therapy, and there are other signs of improvements in biomarkers of hemolysis, which gives us a sense that the bone marrow is returning to a somewhat normal state of function. The 206 trial is the U.S. trial for sickle cell disease. This is a trial that we first treated our first patient in June of 2015. And so that trial is obviously open for enrollment, 8 subjects, and we're looking at a variety of endpoints. We also, as I mentioned, have CCLD. This is a very severe genetic disease, affects boys. It's lethal within short periods of time of showing symptoms. We have great data from an earlier product. This is the trial with a new, more potent, pure product, and we expect, again, to announce data in the first half of 2016 on this program. We also have a pipeline behind that. So I mentioned the TCR program, the oncology program and the immuno-oncology business that we're pursuing. Our lead program in the CAR-T world is partnered with Celgene, and it's targeting BCMA. There was a great article recently in Immunology that articulated why BCMA is a really exciting target for immuno-oncology and CAR-T. And it really is anchored in the fact that it's very specific in where it's expressed, plasma and certain B cells. So the off-target effects are potentially limited in this patient, or in this disease. We're obviously pursuing this in multiple myeloma. Celgene is one of the world leaders in multiple myeloma. Here's some early preclinical data we've disclosed to show why we're excited about this. So in this case, we're looking at animal model of tumor volume over time. And what you can see in the control mice, obviously they progress. In the positive control, you see Velcade having a significant effect, but wanes over time after the therapy is removed. But with one treatment of BB2121, our lead product going into the clinic, you can see very rapidly 2 things happen. The tumors disappear, and you get 100% survival. So this is really exciting data, and hopefully this can be shown to be the case in the clinic as well. Lastly, we have a couple of programs that I'll touch on briefly. We announced a deal with Kite on TCR, so we're moving into the TCR world. In this case, we're targeting HPV-16. It's a next-generation program for us in Kite, where we bring our gene editing technology and our lentiviral manufacturing capabilities merge with Kite's TCR capabilities, and so that's in early stages of development. It's the first time we're really showing the capabilities and the leverage of our gene editing platform, which is anchored in MegaTiles. And the reason we think MegaTiles are interesting and the reason we acquired them was that they're naturally occurring, they're highly specific and efficient, and they're much smaller in size, so you can multiplex. So that technology continues to grow within the company. As an organization, because of us staying ahead of the curve on financing what is arguably, as we all know, a very expensive business to run, especially when you're moving into late-stage clinical and starting to build a commercial infrastructure, both on the manufacturing side and on the commercial side. But we feel like we're in very good position. As of mid-year, we had nearly $1 billion in cash in the bank. And as a company, we've really gone from the bottom of this chart to the top of this chart. The bottom was really about foundation building. It was capabilities, it was experience in the clinic, understanding the patient community, connecting with the patient community and physicians, and then ultimately generating early clinical data. Now we're in this big box up top. And this big box up top is all about trying to become a commercial company. So advancing the data, generating more data, more robust data across multiple clinical programs, building that pipeline behind it, and commercial partnerships for sure in the future, as well as trying to move toward that truly integrated company. And that's the stage we're at right now, and that's what we're going to be building over the next several years. So I want to just thank you for your time.